Now we'll consider how we actually copy or replicate DNA. It's so important that this process goes almost perfectly because this is how we preserve the genetic code and pass it on to the offspring of each of these microbes. And the process has to be completed during a single generation time. It requires the actions of over 30 different enzymes. Don't worry, we're not going to cover all of them, but we will cover some of the major players. The general scheme is we have to separate the two DNA strands, copy the template, and produce two new daughter molecules. Before we begin looking at the details, let's take a quick look at a great animation.
DNA replication is the process in which one double-stranded DNA molecule is used to create two double-stranded molecules with identical DNA sequences. For this process to occur, the two DNA strands are separated, and the DNA replication machinery is assembled at the replication fork. The two DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce two DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semiconservative DNA replication, because half of the DNA molecule is conserved from the original molecule.
So I did say there were over 30 enzymes involved in DNA replication. These, however, are the ones that you'll be responsible for understanding. We'll look at helicase, how it unzips the DNA helix. We'll look at primase, how it synthesizes a primer. DNA polymerase III is our main enzyme that's involved in adding the new nucleotides to the DNA chain. And proofreading for mistakes. DNA polymerase I is going to remove the primer. Ligase will glue it all together. You may be familiar with some of these from your general biology course. Let's begin. The animation just showed us that replication is semi-conservative. That means that each strand acts as a template for the new strand, and each old strand pairs with one new strand. This semi-conservative means of replication allows for accurate replication. It begins at the origin. The origin of replication is a short sequence that's very rich in adenine and thymine. We didn't cover this, but adenine and thymine are only joined by 2 hydrogen bonds, whereas guanine and cytosine are joined by 3. 2 hydrogen bonds are easier to break than 3 hydrogen bonds, so it makes sense that the origin of replication would have lots of A's and T's. It makes it easier for the enzymes to get in and open up a replication fork. So the origin of replication is simply where replication begins. Helicase is the enzyme that's going to bind to the DNA at the origin of replication. Helicase's role is to break the hydrogen bonds between all of those A's and T's and separate the strands. Now, once helicase has done that, there are single-stranded binding proteins that hold the replication fork open at the origin of replication. Before anything can happen, we have to have a primer. We need a primer because DNA polymerase cannot begin synthesis unless it has a short section of RNA to grab onto. It sort of needs a handle in order to get going. Otherwise, DNA polymerase doesn't know where to begin. So, primase is the enzyme that's going to add this primer. Later, the primer will be removed and replaced with DNA nucleotides. Because the bacterial DNA is circular, opening it creates 2 replication forks. So here we can see the origin of replication. That's simply where helicase binds and opens up the 2 DNA strands. As those 2 strands are opened, we can see that there are 2 forks. These are our 2 replication forks. Before we can begin replication, we need to have a primer molecule laid down in order for DNA polymerase to grab on and be able to synthesize the new strand. Now let's take a look at an animation that'll show us how DNA replication proceeds. Don't worry, we'll look at it again as a follow-up to make sure that we understand it.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay, so if you're not familiar with the process of DNA replication, you don't recall it from general biology, perhaps because it was so long ago, or you just didn't get it to start with, this may seem like a really overwhelming process. You just heard about a lot of different enzymes. But let's break it down. First of all, let's look at DNA polymerase III. It's the enzyme that's involved in elongation of the growing strand, but it has some certain conditions. First of all, nucleotides that need to be read by DNA polymerase III are buried in the double helix. So first of all, it has to be unwound and the two strands separated. We've already introduced the idea that helicase does that, and the single-stranded binding proteins hold it apart. But DNA polymerase III is unable to begin synthesizing a chain of nucleotides. It can only continue to add nucleotides to an already existing chain. That's where the primer came in that we previously have discussed. The third component is that DNA polymerase III can only add new nucleotides to the 3' end of a growing strand. You'll recall we were talking about the 3' OH end and the 5' phosphate end when we looked at DNA structure. In this case, the template strand, let's call this the 3' end and the 5' end of the blue strand. DNA polymerase jumped on here and it was reading from 3' to 5'. So reading the blue strand in that direction. Because we're making a complementary strand, and DNA has an anti-parallel structure, the new strand then must have 5' on this end and 3' up this end. So consequently, the new strand is synthesized from 5' to 3'. That's this strand on the right-hand side, the red one. So again, to review that, DNA polymerase can only read from 3 to 5, and thus the complementary strand it lays down is synthesized from 5' to 3'. This results in a neat situation. Because it's only able to read the template from 3 to 5, and thus synthesized from 5 to 3, only one strand can be synthesized continuously. So again, looking into our origin of replication and our 2 replication forks, let's take a closer look at how this works out. So down here, our blue strand is 3' to 5'. So DNA polymerase has to move in this direction along the strand, and synthesis happens in this direction along the strand also. At the origin, we have a primer laid down, and DNA polymerase can get on this strand quite happily and read it from 3' towards the 5' end. In this fashion, it can then synthesize the growing strand from 5' to 3', this being the 5' end of the growing strand, and this being the 3' end of the growing strand. However, in the opposite direction, we have a little bit of an issue, because DNA polymerase, remember, can only read from 3' to 5'. So as the strand very first opened up, only this much was available, and DNA polymerase was reading from 3' to 5' on the blue strand. And it was able to lay down nucleotides from 5' to 3'. Now the strand opened up a little bit more, DNA polymerase was able to jump back, and it would synthesize another piece of strand, reading 3 to 5 and synthesizing 5 to 3. And so now we have a section of new DNA. These little sections are called Okazaki fragments. The strand which can be synthesized continuously, that we saw here, is called the leading strand. And the strand that's being discontinuous in synthesis is called the lagging strand, because it lags slightly behind the leading strand, because it has to be synthesized in fragments. These fragments are called Okazaki fragments. Now, of course, the same thing is happening on the other side of the replication fork. The 3', 5' orientation of the template strand. DNA polymerase can only move this way, which means that it can also only synthesize DNA in this direction. The only difference in terminology of being 5' to 3' occurs because of the orientation of the newly growing strand. This piece is now going to be the lagging strand. And over here, we have a leading strand. If this is new information to you, take a moment thinking about it, working it out. And now let's take a quick look at another animation.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the 2 complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA Primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
So as replication proceeds, the newly produced double strand is going to loop down. DNA polymerase I is going to remove all the primers, as we just saw in that last animation, and it'll replace them with DNA. When the forks come to a full circle and meet, enzymes called ligases move along the lagging strand to connect those Okazaki fragments. In addition, these ligases will complete the synthesis and separation of the two circular daughter molecules. So now we have two circular daughter molecules to move into the new daughter cells. This figure gives a great summary of what's going on in DNA replication. The animations, however, I think really do us justice. There's many of them to check out in the video section of your library in Connect. Now, what happens if there's some incorrect bases added on that growing stand? And this does happen. They say every 10 to the 8th or 10 to the 9th bases, so that's not very often. But these errors occur, and they're usually corrected. If they're not caught and they're not corrected, then they're considered mutations. We'll spend some time later on in the chapter looking at what mutations mean. Basically, we're creating new heritable changes in DNA that are the foundation for possible evolution or advancement of the situation. The cellular integrity is highly dependent on accurate replication, so cells have evolved their own proofreading function to make sure that errors are very few and far between. Both DNA polymerase III and DNA polymerase I can proofread and repair damage. Do you recall which enzyme had which role? Which is the primary DNA polymerase involved in elongation? You're right, DNA polymerase III. And then what did DNA polymerase I have to do with everything? Right, DNA polymerase I replaces the primers. So I have a little bit of a funny representation of what DNA polymerase looks like. I see DNA polymerase as a little van, kind of like a postal truck. And I'm not a great artist. And this postal truck has a tailgate on it. This is the front, in case you can't tell. There are 3 guys that ride in this truck. There's a guy that rides in the front of the truck, and he reads the template strand. And he calls out to a guy who lives in the back of the truck. He calls out the code. The blue guy says, "I see a G." And then the green guy, who's in the back of the truck, leaning out the side door, throws out the complementary nucleotide, which is a C. The guy in the front says, "I see an A." Green guy throws out a T. Guy in the front says, "I see a T." Green guy throws out an A. Every once in a while, they get it messed up. A pretty girl walks down the tracks. The guy that's reading doesn't read it right. The guy in the back's not paying attention. Oops, they threw out a misspelling. So, DNA polymerase III has a third guy who sits here riding on the tailgate. And he reads the letter pairings as the van goes by. And if he finds a messed up reading, then he calls it out. The guys back up. They reread, replace, and move on. So DNA polymerase drives in a 3' to 5' direction all the way down the track, reading and throwing out nucleotides onto the growing strand, growing it from 5' to 3', anti-parallel. The guy in the back proofreads. If they make a mistake, they stop the van, replace the letter, and then move on down the strand. Anyway, this van is my analogy for DNA polymerase III. DNA polymerase I can also proofread, but it doesn't do it as it's elongating a strand. It essentially roams around proofreading DNA to make sure that everything's in place. So there are 2 mechanisms that are evolved to make sure everything goes right. Before we move on, let's take a look at an animation about how repairs happen.
Although mistakes can happen during DNA replication, they are extraordinarily rare. A key reason for this is the proofreading function of DNA polymerase. Roughly 1 out of every 100,000 to 1 million bases attached to the 3' end of a growing DNA chain will be a mismatch, improperly base pairing with the complementary strand. When such a mismatch occurs, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the 3' end with the mismatched base to enter a 3' exonuclease site on the DNA polymerase. The exonuclease site catalyzes the removal of several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown here, though additional nucleotides would actually be removed. Following removal in the 3' to 5' direction, DNA polymerase can then resume its synthesis activity in the 5' to 3' direction. The proofreading function reduces the overall error rate to as low as about 1 in 100 million for DNA polymerase III.
And now before we go, a quick concept check. In Clostridium tetani, DNA is 14% G. What percent of that DNA is A? Gosh, I love this kind of question. So, what do we know? If there's 14% G, and G's match with C's, then there must be 14% C also. When we add these two together, we've taken care of 28% being C's and G's, which means that we're left with 72% split between A and T. And if A's and T's are always paired together, then it's half and half. So we have 36% A and 36% T. So C, 36%, is the correct answer here.